But if you're going to begin to impact the therapeutic space and be invited to speak at the Koch Institute Symposium, you need to realize that you're going to only go so far if you're in the microorganism. So can you, in principle, go from these little guys, lower organisms that have been sequenced, to higher organisms that have been sequenced? So can you move synthetic biology from microbes, which is where the dominant effort has been and still is? And we took that challenge and thought about designing a mammalian toggle switch that could flip between. We thought there'd be whole hosts of applications. It turned out it was really hard. We burned through 2-3 postdocs. Nobody could get it to work. And the challenge there for the dynamicists in the audience who can appreciate it is that in order to have a good toggle switch, you have to have a very tight off. And it turned out the two major ways you could do it would be either with a repressor or with RNAi. And each of those, maybe you get 85, maybe 90% repression. That wasn't enough on the mammalian side. So with Tara Deans, who was a really talented PhD student, she came in the lab, took up the challenge as now victim potentially number 4, and said, okay, instead of doing the toggle switch, why don't we just try to do a really tight mammalian switch, basically half of the toggle. And we're engineers. Let's actually try to harness the two key ways that we could use to shut things off. Can we use both the repressor and the RNAi together? And with that, Tara kind of sat back and came up with a cute design captured here schematically. So she uses LAC to basically shut down transcription that's going to now control the desired expression of whatever gene of interest, in this case it's EGFP. And then what she did was use RNAi, so use short hairpin RNA component that was engineered to target a little target that you tag onto your gene of interest. So you're going to knock down transcription, but if you have a little leakage, this guy's going to knock down the leaking mRNA, and you should, in principle, get a very tight off. Now, we can all get tight offs. You do your knockouts, et cetera. What you want to do is be able to flip the thing on. And so with this design, she also could flip it on with induces IPTG, which will now, basically in this case, shut down LAC, which now will allow TET to come on to shut off the siRNA component. So did this work? Well, Tara got these going in different mammalian cells. Here's some interesting kind of component analysis she did. In synthetic biology, we'll often do component by component. So here's our background. Here's our positive control. When she used the repressor alone, she gets about 90% knockdown. Good, but not great. With just the RNAi, she gets about 85% knockdown. Again, good, not great. When she combines the two into her switch, she gets greater than 99.9% knockdown, which is really good. She can actually flip this thing from an off to an on state and cycle it back and forth. And the beauty is she can actually use this as a rheostat. So like a dimmer switch in your dining room, she can go from effective off and tune it up to different levels and keep it controlled at that level. Of further interest and value is this is, again, a highly modular. The beauty of these engineering approaches in synthetic biology is you can swap in and out different components. So you can put in different promoters. You can put in and use it to control different genes. So Tara wanted to, in kind of a demo of machismo, show how tight her switch was. So what she did as her first demo outside the GFP was she used it to control DTA. So DTA will produce diphtheria toxin, which even the notion is a single molecule of which could kill off a cell. So what she did was create stable cell lines with the switch in the off state, grew them up happily, they were there, and she then could flip them on and selectively kill off the cells that she flipped on, which now opens up an interesting tool for different animal model studies. That is, you can have now different tissue-specific promoters using to drive the expression of DTA, have it in your animal, grow it up, and then kill off certain nerve types or different tissue types to model either aging or disease progression. Another key feature on this is that it's now kind of taken for granted, but the idea really up until the past was just you generally thought of genes as being on or off. But as many in this room appreciate is that many interesting biological processes, and in particular many interesting diseases, are really threshold-based. That is, at a low level you're fine, or maybe at a high level you're fine. There's the intermediate, either it's getting knocked down past a critical threshold or being knocked above a critical threshold. And this switch that Tara's designed is beautifully designed as this rheostat that allows you now to explore different phenotypes. And so what she's showing here is she uses it to control the expression of BAX, a pro-apoptotic gene, and actually shows that at low expressions you don't get apoptosis. And there is some critical threshold above which you actually have now apoptosis being triggered. This switch is now available, I think it's through Origin. It's actually being used by about 800 different groups, which we're really thrilled that it was picked up. In synthetic biology, in addition to these constructs, as I said, really the first part in many areas is to do computation. And we've been excited with Xiao Wang, a postdoc in our lab, recently on looking at can you use computation and eventually synthetic biology techniques to look at issues of multi-stability. And that is how you can have multiple stable states and transitions between those states in regard to different biological processes and also disease. So in stem cells, you have the idea of either both pluripotency and differentiation where this multi-stability is playing a role. And increasingly in diseases, including cancer, there's discussion of could you have certain aspects of the disease arise as your system jumps between stable states. So what Zhao did along with Mo Khalil in our lab was take up this challenge, model starting with just a very simple analysis of a 3-gene, fully connected triad. Okay, so you have 3 genes, could be transcription factors, each talking to each other, so they're interacting with each other, and also regulating themselves. And what he did was a really beautiful design of an in silico high-throughput screening at a qualitative level where he considered all different possibilities of, is this going to be activating or inhibiting, is this going to be activating, inhibiting, et cetera, et cetera. Considered 104 different possible arrangements on the network, and then analyzed using mathematical tools what were the networks that could sustain the greatest possibility of multi-stability. And actually had a number of key ones that popped up. The one that showed the greatest capacity for multi-stability is shown up top. This is one in which each of the genes mutually inhibits the other and activates itself. So you have inhibitory intergene and positive feedback on itself. What's interesting from all those key ones that actually show multi-stability is that Zhao discovered all of them require positive feedback or positive autoregulation in order to have multi-stability. He went further and can actually analyze how do the connection strengths affect the arising of multi-stability. So in a kind of simple table he's showing here, where he's showing here now the strength of the gene-to-gene interaction versus the strength of the autoregulation on the gene. And this is the number of stable states that can arise. So what you see is to get the highest number, which is the highest in this, would be 8 different stable states. And that is, for a given parameter value, it could have 8 different states, right, and jump between it, that you need to have very high autocorrelation, or positive autoregulation, and very low mutual repression. Okay? He then can actually analyze noise strength. So increasingly in the last 5-6 years in synthetic biology and systems biology and now molecular cell biology, there's a lot of interest in understanding how variability, gene expression noise, different changes will allow you to flip between different states. And Zhao can systematically explore that and show that you can actually go, say, have a very stable initial state, and as you increase the noise that could arise from transcription and translation, you'll have your system jump between multiple states. We're now beginning to explore how you could test this. With Kevin Murphy and Gabor Balazzi in our lab, we actually engineered a synthetic gene circuit that will allow you to tune the amount of noise in your system. And what you can do with this is, you can basically ramp up the noise by just increasing gene expression. But what these guys came up with from a synthetic biology standpoint was the ability to change the mean, or in fact, I should say, the ability to change the noise without changing the mean, which now is opening up a number of interesting studies that we're looking at both from disease and differentiation. Switching gears a bit, a few years ago after we began playing the synthetic biology, we were kind of little kids with a hammer looking for nails that we could hit. And so this is around 2002, 2003. We realized, okay, we've got this gene circuit control toolbox, right? A whole series of different switches. We could have cascades, oscillators. What could you do with it? And with Tim Gardner and others, we sat back and returned to our original challenge. Remember when I said when we began thinking about molecular biology, people said, hey, could you reverse engineer the structure of these very complicated circuits inside cells? And now in 2002, we had a lot more data available. We realized, okay, yeah, maybe you could. Right? Maybe you could use these switches as probes to go into a cell, turn different genes up or down, measure the response, and from that infer the underlying circuit. And again, the motivation came from engineering. So if you're an electrical engineering undergraduate, you'll typically take a class, a circuit class, where the professor will hand you a circuit board and say, okay, hotshot, how is this thing wired up? They don't give you the blueprint. Right? So what do? You run your lab, and you turn as many of the components as you can up or down or on or off, and you measure across the circuit board the response. You do this multiple times and try to infer the structure, right? You go back to the professor and say, hey, here's what I found. If it's MIT, you maybe get a C, a D. If you're at Harvard, no matter what you give them, you get an A. And they pat you on the back. Well,